My name is Kevin Tokoph. In this video, we're going to discuss the theory behind the Voges Proskauer test. All right, so let's go over some basics of bacterial metabolism that you may not have seen yet. We know glucose can be converted to pyruvic acid through the process of glycolysis. When we looked at the methyl red test, that pyruvic acid could be converted to mixed organic acids. And if it turned red, it was going to be a positive methyl red test. Now, the Voges Proskauer test is looking at something a little bit different. It turns out that pyruvic acid can be converted ultimately through a series of two enzymes to a molecule called acetoin. Now, acetoin is not directly what we're measuring, but it turns out that acetoin is involved in a series of metabolic transformations. We can form various compounds like diols or diones. Now, if we have bacteria that can produce acetoin, what we can actually do is put in a series of reagents and we can actually convert the acetoin in the absence of enzymes to a dione called a diacetyl. And it turns out that diacetyl can react with peptones, specifically arginine residues of peptones, to produce a red color. Okay, so how do we do that? Well, it turns out that acetoin, if we throw in alpha-naphthylamine, which we've seen before, and potassium hydroxide with oxygen gas, it turns out that acetoin can be converted without the need for an enzyme into this dione called diacetyl. Now, in this mixture, we also have peptones, and peptones contain arginine residues. This is an arginine amino acid that's part of the peptone. It turns out that the arginine can react with the diacetyl, and when it does, we have a color change that is red. Now, basically, what the Voges Proskauer test is measuring is the ability of bacteria to convert glucose into neutral end products. Okay? Diacetyl and butanediol are both neutral end products. However, we're specifically reacting these reagents with the acetoin because the acetoin is indicative of its ability to convert glucose to neutral end products. Okay? And a red color change would be Voges Proskauer positive. Okay? Here's some specific results, what they might look like. Very similar to the methyl red test, if we see usually towards the top of the tube, if we have a red color, or sometimes it can go darker, a little bit purplish, that's going to be a positive Voges Proskauer test. That means that the bacteria are able to convert glucose into these neutral end products such as acetoin and diols and diones. Okay? You can see a positive result right here as well as over here. If we don't see that reddish purple color change, such as over here, or on this side over here, that's a negative result. And so the bacteria do not have this ability. Okay? And also understand this because it can help you tie things together when you're tested on it. The results of the Voges Proskauer test, this red color change that we typically see towards the top, is very similar to the results of the methyl red test that we looked at in another video. So you kind of just remember for Voges Proskauer and methyl red, we want to see a red color for positive for whatever they're measuring, then that might help you a little bit. Although what they're measuring is a little bit different. Methyl red was testing ability to convert pyruvic acid to mixed organic acids, so acids, whereas Voges Proskauer is measuring the ability to convert glucose into neutral end products rather than acidic ones. Okay? And again, I'll mention this, as I've mentioned in the previous videos, this is another one of the techniques that we're going to use to differentiate between gram-negative bacillus. Okay? Make sure to watch the demonstration video on the Voges Proskauer test.